Wow, virus-like proteins giving sight. That is so cool. Can we engineer a new and better virus? David Liu's lab coming out with engineered virus-like particles for efficient in vivo delivery of therapeutic proteins. I thought this was really cool because it actually allows them to use a virus-like approach to actually deliver therapeutic proteins and use the virus's specificity and their ability to target specific cells to actually deliver therapeutic proteins in this case. So this is his Twitter thread about it, and then we can go over the actual paper. So today we report in Cell as engineered virus-like particles platform that mediates efficient in vivo delivery of therapeutic proteins. In vivo delivery means, in vivo means inside, so inside the organism, including base editors. So they're looking at changing nucleotides there, and Cas9 nuclease. So Cas9 nuclease is the scissor basically of the CRISPR. So you have the guide RNA that's going to be finding specific parts of the genome, and then you have the Cas9 nuclease that can actually make those cuts, resulting in therapeutic editing in multiple mouse organs. So the in vivo delivery of proteins into mammalian cells, so we're working with mammals here specifically, remains a key challenge that limits the impact of gene editing in macromolecular therapeutics. So they're saying that the proteins, how do you deliver proteins? So if this is something where you're not just delivering genes to the organism for the organism to make it, you're specifically already making proteins, and then you're delivering those already made proteins. So that's a key difference. Once those proteins get in the system, then of course they can be doing gene editing or for therapeutics, like, well, we'll see. So virus-like particles offer a key advantage of viral and non-viral delivery, but have shown some low protein delivery efficiency in vivo. So basically the protein's not getting there for whatever reason. So in here we have like what a normal virus is, the envelope protein, the capsid protein that's protecting. These viruses package and deliver viral genetic material. So they actually have the DNA or the the DNA or the RNA, and that DNA or RNA gets delivered, and then that's what makes the protein. So for example, like SARS-CoV-2 makes the spike proteins from the RNA. So virus-like particles, capsid cargo fusion. So we have a type of protein that's delivered and bundled here instead of genetic material. My question would be, like, how do they get that in there, and how do they make sure, and how do they make sure that those proteins are what they say they are, or haven't been misfolded once that particle has been made? So inspired by previous reported virus-like protein scaffolds, we designed first-generation VLPs that package base editor ribonucleic protein. So ribonucleoprotein cargo, ribonucleoprotein is another word for CRISPR. So ribo meaning like RNA, so the guide RNA, and the protein is the guide RNA and the protein together, complex together. While V1 VL virus-like proteins efficiently edited cultured cells at high doses, they only mediated 2% editing of the mouse liver following IV injection. So a very low cutting efficiency there. So how do they change it in order to make it better? To unlock the potential of the VLPs to mediate efficient in vivo protein delivery, we systematically identified and engineered solutions to 3 major bottlenecks. Cargo release, so how those proteins are actually leaving the virus-like protein, which makes sense, right? Because if they're bound together, then how are they unbinding? Like, what's the mechanism to actually release that cargo? Cargo packaging and localization. So that would be like, how are they actually getting to and which protein are they actually targeting? That's a good question. And VLP, virus-like protein component stoichiometry that reduce the potency. What do they mean by stoichiometry? Engineering component stoichiometry of engineered virus-like proteins. Oh, okay. So how much? So they're saying how much. That makes sense, because if it's not efficient, right, you could add more proteins or you could make the virus more efficient, right? So there are a couple different ways that you could actually improve that system just by having more of your protein inside the virus-like protein, or you could make the virus more efficient at delivering that protein. And maybe it would be the case that you need a very small percentage of your protein to be actually inside the particle. Maybe you need a lot. So to improve cargo release, we engineered a new linker sequence into second-generation engineered VLPs, virus-like proteins, that support efficient and release and minimize the premature cleavage of cargo before its incorporation. So, okay, so they were being cleaved before they found out that the proteins were being denatured somehow. And that would be the worst case, right? You wouldn't want to deliver something that's somehow changed and not able to do the actual job. On the other hand, you wouldn't want to deliver something at the end that's getting cleaved that isn't doing its job too. So there's two ways that it could be getting denatured, before and after, right? Okay. Next, we use nuclear export signals and nuclear localization signals placed on opposite sites of the cleavable linker to engineer cytoplasmic cargo localization, increasing VLP protein loading by 19 times. Wow. This is like a really common strategy. This is the right here, this little thing, it's NLS, the nuclear localization signal. So there's a lot of different little signals that you can actually add to sequences or proteins that basically tells the cell where this protein should be at any given time. So the idea is, what they're trying to do is they're trying to use that idea to put that onto what their virus like protein, to give it more of a chance to get to there, and that definitely did. There's a lot of different, the cell is very busy, right? So there's a lot of different ways and a lot of different places that these proteins need to go. And in molecular biology, it's kind of taking those ideas and that ability and then repurposing it. So a lot of molecular biology is just taking what's happening in nature and then slapping it onto whatever you want to do. You're not really reinventing the wheel, right? If there's been billions of years of evolution in order to make these nuclear localization signals, then why not just use them? So finally, we engineered an optimal eVLP, which we engineered virus-like protein component stoichiometry that balances the amount of encapsulated cargo with amounts of viral structural proteins and protease, finding that too much or too little of either is suboptimal. That makes sense too. If you are adding too much protein, you're going to change the structure of the virus-like protein. If you're adding too little, it's not going to be efficient when it's delivered. And there's different proteases too, right, that can actually take out the protein that you want to be in there. So these proteases, I'm interested in what those are. They might be targeting proteins that would interfere with this. So they're kind of using a defensive tactic. When they get into where they need to go, it can actually be more efficient. Combining these three solutions yielded fourth generation's engineered virus-like proteins that greatly enhance protein delivery potency in mammalian cells, both in vitro and in vivo. In vitro is outside the cell, so like cellular cell culture, and in vivo inside the actual animal, typically resulting in 5 to 26-fold higher gene editing efficiency relative to, wow, that's crazy. That's amazing. What are they doing though? What are they changing? So they're changing Okay, so they're changing some AT to GC. So one of the early differences and changes to Cas9 was basically the ability to single base pair edit. You know, you can do a bunch of different things with Cas9 now. You can actually make it so they don't cut at all, or you can make it so they make a double-stranded cut. You can make it so that they have a staggered cut. And in this case, you're actually no cut at all, but you're changing the base pair from purine to pyrimidine, or other way around. Version 4 engineered virus-like proteins efficiently deliver base editor protein to various immortalized and primary cell types in vitro, including primary human fibroblasts and T cells. Oh, T cell, this is this is one that people have been talking about for a little bit, to actually change T cells themselves. So instead of like giving as another route to vaccination, what if we just change the T cell and put in that information directly? It's kind of like an interesting idea in order to help the immune system. We can also alter the cell type preferences, tropism, of engineered virus-like proteins by using different envelope glycoprotein delivery, high efficiency, a specific tissue choice, and non-viral delivery, reduced off-targeting editing and risk of DNA integration. That is so cool. And very impressive. Very impressive. Here's the paper. So published in Cell, very prestigious journal. Highlights the engineered virus-like proteins overcome 3 bottlenecks of protein delivery. DNA-free virus-like proteins efficiently deliver gene editing proteins with minimal off-target editing. I guess how would they show that? How did they show that? Did they do like a genome-wide thing? No detectable DNA delivery from, yeah, I mean that makes sense. Minimized off-target editing compared to viral delivery. How did they look at those? Did they sequence? All right. Here's the summary. We're not going to read the whole paper, but we can read the abstract. So methods to deliver gene editing agents in vivo as ribonucleoproteins, so again, CRISPR-Cas9, could offer safety advantages over ribonucleic acid delivery approaches. So nucleic acid delivery approaches like we were talking about, DNA, RNA, that's going to have problems with integration. It can mutate over time. It could not make the same protein over time. It could be silenced. It could be integrated into the genome. There are a bunch of weird problems that could happen with it. We report the development and application of engineered DNA-free virus-like particles, that efficiently package and deliver base editor or Cas9 ribonucleoproteins by engineering VLPs to overcome cargo packaging, release, and localization bottlenecks. We developed 4th generation engineered virus-like proteins that mediate efficient base editing in several primary mouse and human cell types. It is so cool. Using different glycoproteins in engineered virus-like proteins alters their cellular tropism. Single injections of eVLP into mice support therapeutic levels of base editing in multiple tissues, reducing serum, following 63% liver editing, and partially restoring visual function in a mouse model of genetic blindness. I think that is so cool. Restoring visual function in a mouse model of genetic blindness. I mean, that's a great example of how gene therapy could actually be a huge benefit. You know, people kind of talk about the dangers of gene editing, but man, that is such a powerful tool right there. In vivo and in vitro, so outside of the organism in cellular culture, and in vivo, off-target editing from eVLPs was virtually undetected. An improvement over AAV, so lentivirus, and or plasmid delivery. These results establish eVLPs as promising vehicles for therapeutic macromolecule delivery that combine key advantages of both viral and non-viral delivery. How do they make the particles? Production and purification. eVLPs are produced by transient transfection of Jessical producer T293 T cells. Jessical cells were seeded in T75 class as the density for over 24 hours. They were transfected using jet prime transfection reagent for producing the version 1 to version 3 VLPs, a mixture of plasmid expressing VSV. Yeah, but we want the CRISPR, and there you go. And S guide RNA is for the for CRISPR. So they would have to have the RNA in there and then the protein, but how are they gonna where'd the Cas9 come from? They have no detectable nucleic acids, so there's no DNA or RNA after that much time. So the plasmid must be making a DNAs. Oh, okay, so yeah, high throughput. They did do next generation sequencing to check that stuff for off targets.